Well, I'd like to thank the Susan Komen organization for the opportunity to present to you today. Today, I'm going to talk about targeting BRCA1 and BRCA2 mutations and DNA repair abnormalities generally in breast cancer. And this is work done not only in my laboratory at the Cancer Institute of New Jersey, but represents work done by a lot of our investigators in the U.S. and in the world. And so, to give a little background, and invasive breast cancer, unfortunately, is a common problem. And what I want to emphasize here is that treatment of even early stage breast cancer is really targeted against micrometastatic disease. So breast cancer is one of the diseases where even early stage diseases often already has spread beyond the breast. As you all know, the treatment of breast cancer, in fact, most cancers, is divided in kind of two parts. One is local treatment, often involving surgery and radiation, and is basically aimed at getting rid of the tumor in the breast and the draining lymph nodes. And then there's systemic treatment. This is therapy that goes through the whole body, trying to target tumor cells wherever they are. And the thing we've learned early on is that even in early stage breast cancer, it's a small tumor that has no abnormal lymph nodes, we completely take it out and we do nothing else. There's a chance that over the next 5 to 10 years, the disease comes back in other parts of the body. And the idea is that even these small tumors, there's a chance that cells have spread from the primary tumor to other parts of the body and are sitting there like seeds that may take root and grow if left alone. And that's the rationale really for systemic treatment, therapy that goes through the whole body to try and target those micrometastatic cells and prevent them from growing. And in early stage disease, we often use a combination of both local treatment and systemic treatment. In metastatic disease, systemic treatment is often our priority, our main modality. In early stage breast cancer, we often sit there and goes, okay, how can we determine what's the chance that the disease is already micrometastatic and will come back in the future, and identify what we call the metastatic risk of even early stage disease. And for the most part, our risk was defined really by stage, or basically anatomic assessment of how much cancer you have and how much it's spread. And even though they're very complicated staging systems, and this is not the actual staging system, but kind of a little a summary of it. Imagine what happens is early stage diseases have small tumors, and as the tumors get bigger and involve more of the local regional lymph nodes in breast cancer, the axillary, the lymph nodes in the underarm, you start to get higher stage. All right, so stage 1 often are small tumors with no local regional involvement, and as the tumor gets bigger and involves more local lymph nodes or starts to involve other local organs, you start to get higher stage. And ultimately, if there's disease outside the draining lymph nodes in other parts of the body, you consider stage 4 metastatic disease. And what we learned is that, again, even in stage 1 disease, a small primary tumor that's lymph node negative, if you follow women who've just been treated with surgery alone, this is old data from our early trials from the NSABP, you can see that even these small lymph node negative tumors, in both tumors that are considered estrogen receptor negative or estrogen receptor positive, up to 30% of women can have disease come back, or perhaps more, over time. And what's also interesting is that ER positive breast cancers also have a similar rate of recurrence. They just take longer to get there. And those late recurrences are still, early started when the tumor was first taken out. So it's not like ER positive tumors are any less metastatic than ER negative tumors. It's just that the dynamics of the recurrence and how they grow out are different. Right? And the way to think about it in some ways is that, an early stage breast cancer is kind of like finding one dandelion that's gone to seed in your yard. You could take it out, all right? But there's a reasonable chance that next year you're going to find dandelions popping up everywhere. Whereas we think of metastatic disease kind of like is your lawn filled with these. But even here, you realize that you have to not only take out the dandelion, but treat the yard as well to try and prevent these them from filling your yard in the next couple of years. All right. And so even though we talk about metastatic disease, even our treatment of early disease is really aimed, the systemic treatment is aimed at micrometastases. And one interesting thing about this is that you imagine that in early stage disease, how women do and stay alive without cancer depends on stage, how much cancer they had at the beginning. So stage 1, patients with stage 1 disease have less likely disease coming back compared with a more advanced stage tumor. What we've learned is that when we give systemic treatment, we can change the dynamics we can increase the number of women who stay alive without cancer coming back. But the benefit is proportional to the risk, all right? So for some, is an observation we've seen long way. So if you give systemic, for example, chemotherapy, what it can do is sometimes it's cause like a 20% decrease in the chance the tumor comes back. Okay, and even the proportion is the same. The absolute benefit varies by your underlying risk. So, for example, let's say you're stage 1 cancer, and this is a cartoon. This is of any cancer. You can say that. Let's say in here, with surgery alone, 80% of women do fine, but 20% are, in 100 women, 20 women have the disease come back. If the treatment reduces the chance of the disease coming back by 20%, this means that the treatment, in women who have got treatment, four additional women, in addition to the 80 that have done okay without treatment, will now be alive without disease. All right? Whereas now, if you, let's say, more advanced stage cancer, now let's say only 20% of the women will be alive without disease, and 80% will have the disease come back. Now that same 20% reduction in relapse will now cause an additional 16 women to be alive. And so this the absolute magnitude looks greater. And this is important to know because in order to see the kind of benefit in stage 2 and 3, you can see that in a randomized trial in which several hundred women are in each arm of the trial. Whereas to see the kind of benefit in early stage disease, you have to have trials that engage thousands of women to see that benefit. So in many cancers, we still use kind of stage as an idea of when to give treatment. Right? But obviously, this blurs two important things. This is totally ignoring biology. Okay, this is treating breast cancer like one disease, and we know for a long while that breast cancer is not one disease, and different treatments may have different impacts on different subsets. And we learned that early on in breast cancer with estrogen receptors. So early on, we thought that when we developed tamoxifen, we again assess risk just by stage. And early on, we thought, hey, in early stage breast cancer, there's no benefit for adding hormonal therapy because when we did the trials, we can see much of a benefit. But then we got an assay that says, hey, breast cancer isn't uniform in terms of how it responds to hormonal therapy. And in fact, 60 to 70% of breast cancers, express the estrogen receptor, called an estrogen receptor positive disease. And another 30%, don't express the estrogen receptor. And if we just split the tumor by that, we immediately see one thing. Hey, the estrogen receptor positive tumors actually have a better natural history than the estrogen receptor negative. Right? But now if you treat with tamoxifen, all the benefit is seen in the estrogen receptor positive women, and there's no benefit, and maybe even some harm, to the estrogen receptor negative patients with estrogen receptor negative disease. All right. So this says that, hey, you can see benefit if you understand which subsets of disease actually respond to the treatment you're giving, even in early stage disease. And this is a cartoon. This makes obvious sense to us now. These are the actual clinical trial data from NSAB B14 that demonstrated this. And this was the first trial that said, hey, you can define a class of breast cancers that benefits from a treatment, not based on anatomical stage, but based on a biological characteristic of the disease, here estrogen receptor status. All right. But since that time, we still need to do better to identify which breast cancers really benefit from the different treatments that we have. For example, here's one of our landmark trials that was done a while back, in which patients with early stage breast cancer that had involved the lymph nodes, so this is at least stage 2, stage 3 disease, okay, were treated with either standard therapy at that time, which was 4 cycles of chemotherapy called adriamycin and Cytoxan, or that same 4 cycles of chemotherapy with now the new drug, paclitaxel or Taxol. All right, so doubling the chemotherapy, adding a different agent. And when we hear the results of the trial, so this is what we call disease-free survival, which shows plots the number of women alive without disease recurrence over time here in years. And you can see that if you look at the women who got adriamycin Cytoxan alone, 72% of the women were alive without disease at 5 years. Whereas we added double the chemotherapy, now 76% are alive. So in 100 women, an additional 4 would be alive without disease by adding the paclitaxel. Now the question is, okay, is this a stochastic event? There's a random chance that some women benefit, or is there some biological reason where some women benefit from this, others are actually maybe harmed by this, and the net result is that you get this 4% increased survival. And can we find a biological axis to predict that, similar to how we did with estrogen receptor, with hormonal therapy and estrogen receptor status? And so, cool thing is that over the past several decades, we've realized that, hey, breast cancer is not one disease. Breast cancer is a category that probably involves 5, 10, maybe more distinct cancer classes that have distinct underlying biologies, natural histories, and likely responses to treatment. So this is early work from Chuck Perot's group, which showed that using gene expression profiling, that you can subdivide breast cancer at least 5 different subtypes, Malay, basal-like. And this is just a cartoon. Our understanding of this kind of subclassification has grown tremendously over the past decade. But to sit and goes, hey, to really figure out what's working best, we can't treat breast cancer as one disease anymore and really say, hey, what kind of breast cancer do you have? All right. And this kind of understanding is real advanced, that we can identify the natural history of different breast cancer subtypes, identify risk factors for different subtypes. For example, hey, does endocrine factors that are traditionally associated with breast cancer risk, do they only affect the luminal breast cancers, but not the triple negative breast cancers? And ultimately, we can, we're starting to really use these subclasses to do clinical trials in which we target these different subclasses. That's going to be talked about by some of the people talking later today. But the other thing is, of course, gene expression profile and these kind of analyses can be very difficult to do clinically. The platforms changes, and perhaps, they're even easier markers, like even combinations of our classic markers, HER2, ER, PR, and grade, that might be just as helpful. In fact, we're starting to see this. For example, if you reanalyze this old trial, so this is the trial we showed where addition of paclitaxel to their standard chemotherapy had this small benefit. If you now take the same group of patients and split them up into patients whose tumors were ER positive and HER2 negative, which represents about 60 to 70% of tumors, you can see the addition of the paclitaxel had no benefit. Whereas now, if you look at the triple negative group, there was a significant benefit from adding paclitaxel, and also in the HER2 positive group, a significant benefit. The triple negative and the HER2 positive each represent about 15%, and this represents 70%. So together it looks like this, but when you deconvolute them, you can see who's actually benefiting. All right? And so with these kind of approaches is really important, because you remember that our systemic treatment for early stage breast cancer and also metastatic disease is targeting these little cells that have escaped the primary tumor, got into our blood vessels and our lymph nodes, have spread to other organs, and either sit there dormantly and ultimately then grow into tumors that can bother you later. And the idea is, hey, if we now treat with systemic therapy, hormonal therapy, chemotherapy, targeted therapy, immunotherapy, all these things are coming on, we can prevent or delay this outgrowth. But it may be that the effectiveness of each of these components of our systemic therapies depends really on the specific biology of the cancer in an individual patient, in an individual person. All right. And what we often see in our disease is that we first check our therapies in metastatic disease. So, for example, here is early data from using HER2 targeted therapy in a patient with HER2 amplified breast cancer, in which here is a bone PET scan showing extensive disease in the bones and the liver, and the target therapy causes dramatic response. All right. And then once we see efficacy in the metastatic setting, we then take it into the early disease and say, hey, if it can target gross metastatic disease, can it target the micrometastatic disease in early stage breast cancer and improve outcome? And in fact, that's what we see. We first did the metastatic trials, and then we went to the early phase trials and showed that, hey, now if we compare chemotherapy given after surgery versus chemotherapy plus the HER2 targeted agent, here trastuzumab, you can see a dramatic increase in the number of women alive without the disease coming back. All right. And so the advances in our understanding of treating metastatic disease ultimately translate into our advances in treating micrometastatic disease in the early stage patients. So today I'm going to shift courses from this kind of general background and talk now about the biology of BRCA1 and BRCA2 mutant breast cancer, which are really a subset of breast cancers that arise. As you know, breast cancer is a common disease now. Over about 300,000 women each year in the U.S. develop breast cancer, and about a small percentage of these, about 5 to 10% of these, arise in families with a high incidence of breast cancer and may be associated with a familial predisposition. If you look at these high-risk families, so you look at that 5 to 10%, of those, 15% of them will have that breast cancer risk will be due to mutations in genes called BRCA1 and BRCA2 for breast cancer gene 1 and breast cancer gene 2. All right, another 3 to 4%, another somewhere between 3 to 7% may be due to other genes, which we'll talk about a little later. And for most of these, we either don't know or have complicated models to understand the risk in those. So a small percentage of women are due to due to mutations in BRCA1 and BRCA2, right? And the story of BRCA1 and BRCA2 is really, interesting and very informative. And so, in fact, one of my scientific heroes, Mary Claire King, used old-fashioned genetics. So in the 80s and even the early 90s, people thought most cancer was due to, environmental risk factors like smoking for lung cancer. And so Mary Claire King, from her experience with some of these families, said that, hey, there's got to be in some families underlying genetic risk. And by very careful, old-fashioned, human genetic analysis, she was able to identify a region on human chromosome 17 that's seen to correlate with early onset familial breast cancer risk. And she labeled this locus breast cancer gene locus 1, or BRCA1. All right. Since it was identified, she had put her paper out in 1990, in about 4 years using novel genomic technology, this gene was ultimately identified. And it was found that BRCA1 acts as a tumor suppressor. And the way this works is that, all of us have 2 copies of every gene, one from our mom and one from our dad. All right. And, women or a man with a germline mutation of BRCA1, that means that one of their copies of BRCA1 present in all cells is non-functional or is mute, but the other copy is fine. And cells that just have one bad and one good copy of BRCA1 are fine because that good copy is the backup and everything works well. But the cancers that arise in these BRCA1 mutation carriers have all acquired another mutation in the normal copy of BRCA1. And so now they get both copies of BRCA1 are now mutant, and then that causes cancer. As we call it a tumor suppressor. As long as you have one good copy, it prevents cancers from, it suppresses the development of tumors. All right. Patients who are born with one mutant copy of BRCA1 or BRCA2 have an increased risk because they're easier for them to lose the other copy than someone who has 2 intact copies of BRCA1 or BRCA2. All right. And we learned a lot about the human BRCA1 cancer syndrome. So women carrying a mutant allele of BRCA1 have a greatly increased lifetime risk of both breast and ovarian cancer, about a 50 to 80% lifetime risk of breast cancer, and somewhere between a 15 to 40% risk of ovarian cancer. Ovarian cancer is actually much rarer than breast cancer, so actually the relative risk, they actually have a much greater absolute relative risk of ovarian cancer than breast cancer. The tumors that arise in these women all have lost the wild type, the normal copy of BRCA1, again, implying the BRCA1 functions as a tumor suppressor. And intriguingly, men with a BRCA mutation have little or no increased cancer risk. And if you look at the breast cancers that arise in mutation carriers, they tend to have early age of onset. So if you look at this graph, the bottom line here is the incidence. On the y-axis is the risk of breast cancer, and on the x-axis is age. So this is a normal risk that increases with age. If you look at a BRCA mutation carrier, you see the risk rises much earlier, even in the 30s, and rises dramatically between 30 and 60 years of age. All right. And the cancers that arise are not just any breast cancer, but these high-grade triple negative basal-like breast cancers. All right. And there's no, an interesting thing is also there's no clear consensus on increased presence of preeminent breast lesions in BRCA mutation carriers. So if you look at the prophylactic mastectomy specimen of a BRCA mutation carrier, it's not like the breast is filled with preeminent lesions. So this is not like other familial cancer syndromes like APC, the colon cancer syndrome, where by age 30, your colon is full of polyps, all right, and you inevitably get colon cancer. Or like P53, Lee-Fraumeni syndrome, you get multiple cancers in early childhood. Here you may or may not get a cancer during your a lifetime. And the work that I'm going to talk about next is really, once the gene was identified, the big question was, what does this gene do, and why does its dysfunction predispose to breast and ovarian cancer? And this early work was done by the laboratory of David Livingston, who I was fortunate enough to actually do a postdoctoral fellowship with when I was at the Farber. And the early work was done by a postdoc named Ralph Scully in his lab. And I pull him up because, unfortunately, David just passed away this past year unexpectedly at age 80, and made me think a lot about his work and seminal contribution to our understanding of the field. Right? And so early on, it was thought that, hey, BRCA1, loss of BRCA1 causes breast and ovarian cancer. So it's thought that BRCA1, the gene, would have something to do with the biology of specifically for the breast and the ovary, all right, semi-controlled growth of that. And so to our surprise, when BRCA1 was ultimately cloned and then characterized as a protein, it was found to be this large protein that didn't look like any other gene that had been characterized so far. And its expression was not limited to breast and ovary. In fact, it was present in all cells. And in fact, the thing that tells you when it was expressed or not was when cells started going into cell division. So when the cell started to divide into 2 cells and start to synthesize DNA, regardless of what cell you were, BRCA1 will be expressed. And actually it's present in the nucleus. So where DNA is in a cell is in the nucleus of the cell. As these little this is a fluorescence picture of BRCA1, is these little speckled dots in the nucleus. So it was this protein that comes up when cells make DNA and is present in the nucleus in these little nuclear foci. All right. And the other thing was, noted early on, was that if you take out BRCA1 from cells, the cells don't grow better. they actually rapidly stop growing and develop gross chromosomal abnormalities. All right. So we realized that BRCA1 was important to somehow maintain the integrity of the chromosomes and cells. When BRCA2 was cloned, it was found to also be a large protein and had no homology to BRCA1. It didn't look at all like BRCA1 in terms of its gene sequence or the protein sequence. All right. And so we always say that BRCA1 and BRCA2 are 80% homologous by name, but nothing else. But even though they looked completely different in terms of the gene, they remarkably had very similar properties. It also is widely expressed in all proliferating tissues, was found these little nuclear foci were actually co-localized with BRCA1. And again, if you take out BRCA2 cells, they rapidly undergo proliferation arrest and develop chromosomal abnormalities. So BRCA1 and BRCA2, even though they are different genes, they don't look like each other, they functionally quite related. And the big question is, what do BRCA1 and BRCA2 normally do in cells? There are a lot of answers to that. But the basic thing is that if you lose BRCA1 and BRCA2 and you go through cell cycles, your chromosomes, these are mouse chromosomes that normally are beautiful and paired up and look very regular. If you now allow a mouse cell to go a couple of cell divisions without BRCA1 or BRCA2, chromosomes fall apart. You get these weird structures. And so basically, loss of BRCA1 and BRCA2 normally works to kind of maintain the normal chromosomal structure in cells. All right. This raised the possibility, and this early work was done with Ralph Scully and actually in David's lab when I was a resident in internal medicine at the Brigham. We said, hey, the BRCA1 deficient cells were actually very sensitive to certain DNA damaging agents and may have an underlying defect in DNA repair. And so if you take BRCA1 mutant cells and see how are they sensitive to, for example, ionizing radiation or gamma radiation, they're much more sensitive to radiation than cells that had intact BRCA1. Right? And similarly, if you sit there and go, so how well do BRCA1 mutant cells repair DNA? And there's a way to kind of check the dynamics of DNA repair. You induce a lot of breaks and see how quickly the breaks are broken. So a normal cell, shown in this lower line here, the majority of the DNA breaks are rapidly cleared in the first several hours. And then there's a second phase which lags over the next 24 hours. In BRCA1 mutant cells, what happens is that first phase of repair is fine, but then they lag. They fail to repair the rest of the DNA over the next 24 hours. And this suggested that BRCA1 mutant cells have a defect in repairing DNA breaks. All right. And that may underlie, that maybe cells that lost BRCA1 and BRCA2 now have more mutations because they can't repair DNA, and that's what causes the cancer predisposition syndrome. And in fact, work from multiple laboratories, including the laboratory of Maria Jason, Ekstraman, and others, really clarified the role of BRCA1 and BRCA2 in a specific way in which cells repair DNA. So imagine that this is a cartoon of DNA. If you have a DNA break, two things happen. One is that the integrity of the chromosome is broken. It's literally now in two pieces. The second thing is that information, often because of the base pairs on each side are kind of removed, some DNA information is lost. And the question is, how do the cells repair this? Well, one way they can do it is just basically take the ends with minimal processing, just slam them together. All right, and that will anneal the DNA and get rid of the break. All right, and this is a process called non-homologous end joining or single-strand annealing, and these are highly efficient, but they're very mutagenic because they don't recover the information. You slam them together, and often these are what we call error-prone ways to repair this DNA break. However, in cells that are dividing DNA, they can actually copy off of what's called a sister chromatid. They can copy this missing information from a nearby piece of DNA and recover it. And this is called homology-mediated repair, and this is error-free. And this is a really important pathway of maintaining genomic stability in mammalian cells. And what we found was that BRCA1 is really involved in this error-free way of repairing DNA double-strand breaks during DNA synthesis. And if you lose BRCA1, cells inappropriately use these error-prone ways, and this leads to genomic chaos, increased mutations, and cancer. All right. The important thing is that somehow the cancers that arise, even though they don't have BRCA1 and have this profound defect in DNA repair, they somehow survive. But we realize that now they may have an underlying defect in DNA repair that can be exploited for therapy. And in fact, early work suggested that BRCA1 mutant cells, and this is work done by collaborator of ours, Jas Jankers of the Netherlands Cancer Institute, who created mouse models of breast cancer that were BRCA1 deficient. We found that, hey, they were actually quite sensitive to certain agents like cisplatin and doxorubicin, all right, and less sensitive to certain others like docetaxel, suggesting that that tumors that have underlying defects in BRCA1 may actually be more sensitive to certain classes of our standard DNA damaging agents. right? Similarly, shortly after this, work by Thomas Halliday's lab and Al Ashworth suggested, hey, there's a series of other drugs called PARP inhibitors that also work on DNA that were specifically toxic to cells with impaired this homology-mediated DNA double-strand repair. And basically, where extreme cells that have mutations in BRCA1 or BRCA2 were exquisitely sensitive to this. And these are called these poly-ADP ribose polymerase, or PARP inhibitors. And this was work done in back in 2005, so less than about 10 years after BRCA1 was cloned. There was all of a sudden, from the basic biology, all of a sudden specific treatments that might work on this. And the way the PARP inhibitors are thought to work is that PARP, or poly-ADP ribose, is an enzyme that normally helps the body deal with these what we call DNA nicks. These are not double-strand breaks, but little nicks in DNA that occur all the time from oxidative damage that's going on in our bodies. Okay, and what happens is that PARP normally very efficiently repairs them. If you don't have PARP, or PARP is inhibited by a drug, these little nicks don't get repaired. And again, cells don't care, but if the cell now enters S phase, starts to divide, then these little single-strand breaks are converted into DNA double-strand breaks when the cell starts to divide, starts to replicate their DNA. All right. This again is also no problem, because if BRCA1 and BRCA2 is present in these cells, they do their job and kind of repair this kind of break, and the cell division goes on, and everything is fine, everything is repaired. However, the cells don't have BRCA1 or BRCA2. The cells can't deal with this kind of break, and then the cells get lethal damage and then die. All right, and this is called synthetic lethality. So these normal cells will tolerate the PARP inhibitors and the kind of damage that they induce, but if the cells don't have lost both copies of BRCA1 or BRCA2, now this is lethal. We've got a little bit idea that this may not be exactly how it works. We now also realize, for the more sophisticated, that PARP inhibitors may work in a different way, in that they actually trap PARP on damaged DNA. And so they actually trap the PARP, and this creates these bulky DNA damage. So this may actually be the DNA damage that the BRCA1 or BRCA2 mutant cells can't repair. All right? But this early observation back in 2005 was rapidly converted into a phase 1 clinical trial of this in 2009, so within 4 years. All right, and it shows that these PARP inhibitors, poly-ADP ribose polymerase inhibitors, actually had significant activity in any cancer that had BRCA1 or BRCA2 mutations. So this is a rare thing when you get a phase 1 trial, first in human study, that gets published in New England Journal of Medicine. All right, and it was an unusual phase 1 study in which the entry criteria wasn't a specific kind of cancer, but any cancer. This include ovarian cancer, prostate cancer, and mostly breast cancer, that had a mutation in BRCA1 or BRCA2. And what they saw was that there was these dramatic responses that were seen in these patients with this inhibitor. And it took a while, from 2009 to now 2017, in which we were able to show that the same drug, olaparib, all right, was actually effective in patients compared with standard therapy in now patients with metastatic breast cancer that had a germline mutation of BRCA1 or BRCA2. And again, this is the progression-free survival plotted from patients who are treated with this oral drug olaparib versus standard chemotherapy, showing that this is actually superior to standard chemotherapy. This drug. Again, this took a while. This phase 1 study was done in 2009, and it took till 2017 to get this approved in breast cancer. In addition to olaparib, now and what interesting thing was, if you look at who benefited, almost anyone with BRCA1 benefited, including those with this triple negative BR, which is the majority of the BRCA1 mutant cancers, which are thought to be traditionally tough to treat. They're the ones who had significant benefit from these PARP inhibitors. And in fact, other PARP inhibitors have come in. So talazoparib, 2 or 3 others have come in and have shown to be effective in advanced breast cancer with a germline mutation. What's great is, as soon as we saw the efficacy in advanced metastatic disease, the question was, hey, will this be effective in earlier stage disease in the setting of micrometastatic disease? And so that data just came in the New England, just published in New England Journal of Medicine just this in the past several months. And this showed that, hey, now if you take the same drug, olaparib, the PARP inhibitor, and now use it for the systemic treatment of patients with early stage cancers that arose in BRCA1 or BRCA2 mutation carriers, and treat them after the primary tumor is removed, what happens? You can see that the patients who were treated with olaparib, this oral drug, had a greater chance of being alive without disease when compared with just getting standard chemotherapy and nothing else. All right, and in fact, not only there would be greater chance of being alive without disease, there actually a greater chance of being alive at 3 years. All right, showing that how our understanding of basic biology of BRCA1 led to development inhibitors that affected metastatic disease, and then ultimately also early phase disease. All right. The question is, hey, you see this kind of effect in early stage disease, but if you look at the metastatic advanced disease, you can see that, hey, over time, still most people have disease progression despite being treated with olaparib, whereas in the early phase setting, you can see that there's this prolonged effect. And what's the difference? So one of the differences is that, in metastatic disease, people have literally kilograms of tumor, whereas in the early phase disease, even though we're treating micrometastatic disease, there's probably, hundreds to just thousands of cells that are left. So the disease burden is much less, and there's a greater chance to completely eradicate this. Whereas in the advanced disease, you can develop resistance. There's enough cells that you can develop resistance. And the mechanisms of resistance to these PARP inhibitors and also my chemotherapy dose, we recently figured this out, and is actually very, tells us a lot about the biology of the disease and perhaps new strategies to treat it. So if you take tumor cells that are BRCA1 or 2 deficient and you treat them in culture with platinum, initially you have this great response, but ultimately you can get tumor cells that grow out that are completely resistant. Similarly, if you take mouse models of BRCA1 mutant cancer that normally grow very rapidly, and you treat them with olaparib, initially you see this nice response in the tumors, then ultimately you start to see the tumors start growing out with resistance. All right. So now the question is, how do these tumors become resistant? These BRCA1 to these agents. And so a lot of insight was done here by Alan Ashworth and also independently by Toshi Tamaguchi, in which they took in cell lines, they took cells that were BRCA2 mutant, all right, and treated them with platinum and waited for resistance to occur. And just to remind you, remember that mutations in BRCA1 and BRCA2 are mostly what we call frame shift or truncating mutations that basically cause the so this is the full gene here of BRCA2. There's a mutation here that causes a frame shift, and what happens is now there's an early premature truncation of the gene. And often what happens is, without going into the details, when you have these early truncations, often the RNA is unstable, and these are actually functionally null. So often no protein is produced at all. All right. And so if you take a cell line that has this kind of mutation of BRCA2, you treat the platinum and wait for resistance to develop, and then you sequence the resistant cells. What happens? What the Ashworth group found was that these resistant cells had acquired a second mutation in BRCA2 that excised the frameshift mutation and allowed the gene to come back what we call in frame. And so now it converted this truncated gene to a protein that just had a small internal deletion. So it was a wild thing in which a second mutation of the same gene actually restored function. We call these reversion mutations. All right. And this shows that, hey, the way these cells became resistant to platinum was to restore BRCA2 function with another mutation. Okay, and similarly with BRCA1, you can see here a woman that had a truncating mutation in BRCA1 was highly sensitive to platinum as an ovarian cancer. When she developed resistance to platinum, and they resequenced the tumor, now in addition to her initial deletion of one nucleotide, there was now an additional 23 nucleotides deleted. And so for those who remember the frames of DNA, 23 plus 1 is 24, that's divisible by 3. So now this is converts this truncating mutation into a small internal deletion of 8 amino acids. All right. And now this is functional repair and is resistant. So these secondary mutations actually initiate resistance to the PARP inhibitors. All right. And interestingly, you see these same kind of deletions in resistance to a variety of chemotherapeutic agents. And in fact, these deletions have the features of microhomology, i.e. the inappropriate use of those error-prone DNA repair mechanisms is what causes these secondary mutations to arise and then be selected for. All right. And in addition to that, there are we and others, our group and others, including the group of Andre Nuzensweig and us in combination with the laboratory of Jas Jonkers and Madalena Tarsunis, in addition to reversion mutations, you can actually get other mechanisms of resistance. This is work done by Andre Nuzensweig, a lab with Sam Bunting, and also my lab in collaboration with the laboratory of Jas Jonkers and Madalena Tarsunis, found that another way to acquire resistance to PARP inhibitors in BRCA deficient cancers is actually mutations in other parts of the DNA repair pathway, what we call compensating mutations. Now I won't go into the bloody details of this, but it's kind of funny that mutations in one part of the DNA, that defects in DNA repair caused by BRCA1 can actually be reversed by getting other mutations in other parts of the DNA repair pathway. And one way to think about how this works is that normally, when you have a DNA double-strand break, in order to get error-free repair, you have to have what's called processing of the ends. And we know there's a gene called 50BP1, which is present during all phases of the cell cycle, that basically just prevents the ends from being processed. And if you can't do that, the only thing you can do is slam the ends together and you use this error-free, this error-prone pathway called non-homogeneous end joining. When S phase comes around, BRCA1 is now expressed. And we think one of the functions of BRCA1 is to prevent this gene 50BP1 from doing its function. All right, so this prevents 50BP1 from preventing end resection. So now end resection is allowed. You chew back the ends, and now you can now get these DNA strands freed up for error-free repair. Okay, and you get homologous recombination. Now, if you have cells that lack BRCA1, what we see is that now during S phase, 50BP1 can do what it normally does outside of S phase, and that's to inhibit end processing. And this, again, prevents HR, and now you get inappropriate non-homogeneous end joining occurring during S phase, and you get genomic chaos and sensitivity to PARP inhibitors. But now, if you get rid of both 50BP1 and BRCA1, end rejoining is again released, and now you can have appropriate homologous recombination without no longer sensitive to PARP inhibitors. And so somehow two wrongs make a right. You can get rid, if you get rid of BRCA1, you get this enormous defect in DNA repair. If you get rid of BRCA1 and 50BP1, the defect in DNA repair is now actually reversed, and you're no longer sensitive to PARP inhibitors. And we have shown that it's also a mechanism of resistance to the PARP inhibitors. All right. And so in fact, if you look at kind of how patients get, all the mechanisms by which you can get resistance to PARP inhibitors in BRCA1 and BRCA mutant cancers, there are several mechanisms that we've recently summarized in a review article. So some of these mechanisms are very drug specific. You can have specific pumps that come in that pump out the PARP inhibitors or specific drugs. If that happens, you can actually just switch agents and you can still get a response. But otherwise, you get what's called pathway reversion, in which the underlying defect in DNA repair is now restored. If that happens, you now are resistant to the PARP inhibitors and actually to a lot of the DNA damaging agents. All right, and we're doing a lot of work in how these kind of mechanisms that we call pathway restoration occurs in BRCA mutant cells. Okay, but the presence of these really tells us a lot about the biology of BRCA1 and its role in tumorigenesis. Well, what do reversion mutations tell us? So one is that the presence of these reversion and compensating mutations that arise as mechanism of resistance to PARP inhibitors and likely to platinum and anthracyclines, our standard chemotherapy agents, tell that these agents, our standard chemotherapy agents, as well as the PARP inhibitors, are all targeted therapies for these BRCA1 and BRCA mutant cancers. Because when we treat them with that and they become resistant, they develop these reversion mutations. All right. It also shows that the loss of BRCA1 and BRCA1 function is only required for tumor initiation. You can't cure a BRCA1 mutant cancer by putting back in normal BRCA1, because in fact the tumors are happier. They required that defect in DNA repair only to initiate tumorigenesis. Once the tumor is established, it actually doesn't care about the DNA repair defect anymore. All right. And also shows how the resistant platinum and PARP are kind of correlated. All right. And we're doing a lot of work on really, how to overcome resistance to these resistance mechanisms. So this brings up, because now we have these, techniques that can target BRCA1 and BRCA2, we're now finding BRCA1 and BRCA2 mutations in cancers and other areas where we didn't expect them. And this is due to the rise of, what we call next generation tumor sequencing. So now, tumor sequencing, which used to be only done in cell lines or in models with great expense and time, we can now, in an individual patient's tumor, we can target our favorite set of, 50 to 400 genes and capture that, sequence them to high quality, and get the results back in a week to 2 weeks. All right. And this is often done to better characterize tumors, to identify the mutations that are driving digital cancers and form the backbone of what we call precision oncology. And in fact, at the Rutgers Cancer Institute, we have this protocol we've had since 2012, in which patients with many rare poor prognosis cancers get kind of DNA from both blood and tumors retrieved, and we do targeted sequencing of a bunch of cancer genes and present them on a molecular tumor board and review data and follow treatment and outcomes. all right? And this kind of protocol has been going on at our institute for the past now almost 10 years. And we've gotten a lot of experience in understanding how to deal with sequencing data. And one of the problems that sometimes comes up is sometimes we see mutations in BRCA and BRCA2 in scenarios that we didn't expect it. So I'll give you the original patient that we saw that raised this kind of a problem. There's a gentleman who presented with a kidney mass, and this was resected and found to be a kidney cancer. It has some atypical features, and unfortunately, after the surgery, he had rapid recurrence of the tumor both in the surgical area and also in the lung. And this was treated with the usual agents for kidney cancer at that time, and the tumor either progressed or he didn't tolerate the treatment. And so then he was enrolled in our sequencing protocol, and his tumor was sequenced. And what we found was these were the alterations in his tumor. And in addition to the mutations in F2, CDKN2A, AR1A, there was this mutation in BRCA2. And it's a classic frameshift mutation, and actually seen in some, is known to be a germline mutation carrier. In fact, we sequenced his blood and showed that he, in fact, was a BRCA2 mutation carrier. All right. But now the question is, okay, he's a guy with a BRCA2 mutation, and he develops a kidney cancer. Is this just a sporadic tumor? Just because you have a heterozygous mutation of BRCA1 doesn't mean you're immune to normal cancer, especially a gentleman. Or has this tumor really lost the wild type copy of the gene in the tumor cells? And so this is really a BRCA2 deficient cell, which then would be sensitive to things like PARP inhibitors and platinum, which we normally do not treat kidney cancer with. All right. And can we figure this out? All right. The cool thing is that by using computational methods, here we say this, the allele frequency is 2%, estimated tumor frequency 20%, without going into the details. By using kind of computational methods to tease out the exact kind of nature of the mutation, we can actually estimate whether the wild type allele of BRCA2 is intact in this tumor or not. So this is work done by Hossein Kiabanian, who's a computational scientist at our institute, who developed methods called Logic, or LOH and genomic inference calculator, to look at high-depth sequencing data and say, hey, when you see a BRCA1 or 2 mutation in a tumor, what's the likelihood it's germline, and what's the likelihood the wild type copy is gone in the tumor? All right. And when we use this kind of analysis in this tumor, we actually showed that this tumor, to our surprise, had evidence that, in fact, the wild type copy of BRCA2 was lost in this tumor, and that this is a tumor that would be, sensitive to PARP inhibitors and platinum. In fact, we treated them with a platinum-based regimen and had a great response. All right, and so we realized, hey, when we're starting to sequence tumors, we see this more obviously. We see a tumor, which all of a sudden we see a BRCA1 or BRCA2 mutation, which we didn't anticipate. And how do we figure out whether this is, is just a passenger mutation that just came along the ride, doesn't really join, or is this really BRCA1, 2 deficient tumor? And so we started developing these analyses and started looking at a larger spectrum. Saying, hey, when we see what's the frequency of BRCA1 or BRCA2 alterations in all cancer types, and when we see an alteration, how often is the normal allele gone? Is truly a BRCA2 deficient tumor that might be sensitive to platinum PARP inhibitors? And so this is work we did in collaboration with Foundation Medicine. We looked at a set of about 230,000 cancers that have been sequenced through FMI. And this is work done by Ethan Sokol at FMI and Hossein Kiabani at my institute. And what we found was that if you look at pathogenic mutations in BRCA2 that were sequenced in all cancer types, this is the relative frequency in different cancer types here. And what we can see is that, as expected, we see a reasonable incidence of BRCA1 and BRCA2 mutations in ovary, prostate, breast, interestingly in pancreas cancer, which are known to be associated with the BRCA1 and BRCA2 cancer syndrome. All right, but intriguingly, we also saw an incidence in skin cancer. Sorry about that. We saw an increased incidence in squamous cell skin cancer, okay, as well as in other cancers like bladder cancer, bile duct cancer, even lung cancer, at somewhere approaching 3 to 4%. All right. So initially said, hey, maybe BRCA1 alterations are more widespread than we think. However, when we asked the second question, okay, how often we see these BRCA1 and BRCA2 alterations, do we actually see loss of the wild type copy? And saying this is really, truly has lost BRCA1 and BRCA2 function. All of a sudden, the numbers, almost every tumor that in ovary, prostate, breast, and pancreas, if you see a mutation, almost all of them have lost a second copy. Whereas the rates of loss of the wild type copy in other cancers is much less, but not 0. So instead of being 4 or 5%, now it's like 1% in these other cancers, saying that most of the mutations seen in BRCA and BRCA2 in cancers outside of ovary, breast, prostate, and pancreas are mostly passenger mutations, and the wild type copy is in fact intact, and so would not be sensitive to platinum or PARP inhibitors. All right, and in fact, if you look at this, even patients with germline or somatic mutations in BRCA1 or BRCA2, how often do we see that the second allele is gone? You can see if you have an ovarian cancer, and you have mutation of BRCA1 or BRCA2, almost all the time there's evidence of the mutation being biallelic. Right? Both copies are affected, here shown in blue. Similarly, you see the same thing in breast. If you see a mutation in BRCA1 or BRCA2, the vast majority of cancers, whether it's germline or somatic, tend to have lost the wild type allele of BRCA2 in these really deficient tumors. In prostate, it's very different. If you have a mutation in BRCA2, and you have a germline mutation in BRCA2, and you have prostate cancer, the vast majority of cancers show evidence of loss of the wild type allele, and this says that the BRCA2 is a really prostate cancer tumor suppressor, and these cancers will be sensitive to PARP inhibitors. However, if instead you're a BRCA1 mutant carrier, and you develop prostate cancer, most of the prostate cancers have not lost the wild type copy, suggesting that BRCA1, is not really a prostate cancer gene, but BRCA2 is in this setting. Interestingly, in pancreas cancer, we see evidence of loss of heterozygosity. We have germline BRCA1 or BRCA2. All right, and if you look at other cancers, not ovarian, breast, prostate, the likelihood of LOH is much less. All right, suggesting that only a minority of cancers, even if you see BRCA and BRCA mutations in these other cancer types, is there truly loss of the wild type allele, and that would suggest that BRCA is really playing a functional role in the development of that tumor, and you can use PARP inhibitors. So the next question we asked is, is biallelic BRCA and BRCA mutation associated with evidence of an HR defect and thus sensitivity to PARP inhibitors? And so there's ways to look, can you look at features of the tumor, genomic scars that said, hey, when we see these biallelic BRCA and BRCA, is there really other evidence that there's really this gross genomic abnormality, like we saw those chromosomes all messed up? And there's ways to do it. There's a thing called global LOH score, which can kind of be a surrogate marker for the presence of this kind of total genomic chaos that occurs when you really lose BRCA1 and BRCA2 function. What we found is that if you look at the cancers in which we predicted that there was a biallelic loss of BRCA1 and BRCA2 and compared this global LOH scores to tumors that either had monoallelic loss, only one allele affected, or were wild type, you can see that even in these other cancers like bile duct cancer, non-small cell lung cancer, if there was the small percentage of cancer that had evidence of biallelic loss, there was actually evidence of this underlying genomic catastrophe, suggesting that that small percentage of tumors may in fact be sensitive to PARP inhibitors. So biallelic, so the presence of BRCA and BRCA2 mutation is not enough to consider, but you have to see if there's really evidence of loss of the functioning allele, and that may be a biomarker of PARP inhibitor sensitivity in many cancers, in addition to ovary and breast. The other question then we asked, which we can the other genes that might be involved is that there are other DNA repair genes, in addition to BRCA and BRCA2, that may also function the same HR pathway. And there are some that really function closely with BRCA1, like PALB2, and others that are kind of associated with DNA repair, but not be associated with BRCA1 and BRCA2. And one of the questions we asked was, hey, are all mutations in DNA repair similar to BRCA and BRCA2 in having the same kind of effect and potential sensitivity to DNA drugs like PARP inhibitors, or they work differently? So what we did is, again, working with Foundation Medicine, we asked, okay, when we see biallelic loss of these genes, which of these genes do we see also evidence of this kind of gross genomic chaos? And what we found was that there was strong association for some of the core genes, BRCA1 and BRCA2, but also genes like RAF51, PALB2, and RAF51C. But there's no association with other genes that we also think were associated with DNA repair, like ATM, CHEK2, suggesting that these genes, even though they're considered DNA repair genes, may have very different effect on the tumor than BRCA1, BRCA2, and the core HR genes. And so that not all DNA repair genes can be lumped together as one category. You have to really understand what they do in order to get the optimal treatment for them. All right. But if you look at this, you say, okay, if you look at these kind of core HR genes, how often do you see biallelic mutations in these cancers? You can see in ovary, prostate, breast, surprisingly, up to 5% of pancreas cancer, you can see these biallelic alterations. In other tumors, it's much less. It's on the order of 1% or so. But still, 1% of lung cancer still represents a significant number of people, suggesting, hey, we want to investigate these biallelic alterations in these other cancers to see if these may also benefit from PARP inhibitors, like we've shown in breast and ovarian cancer. All right. At the end, I was talk about, one of the things that we learned from this is that, that some of our classical chemotherapy and PARP inhibitors, which in part work like some of our classical chemotherapy, are really all targeted agents. And if we think of them, in the old days, we consider these, our chemotherapy as kind of gross poisons that we wanted to give as much as possible, all right, maximum tolerated dose in order to get the response, because we thought they were function as non-specific poisons. But now if we think of them as targeted therapies, i.e. they will only work in terms of underlying defects in DNA repair, can we in fact now reconsider dosing? All right, and say, hey, do we have to give the maximum tolerated dose, or can we come up with a less toxic way to give even our standard chemotherapeutic agents to maintain efficacy? And this is work that we've done with Nancy Chan. We started and said, hey, can we do this in breast cancer? And we noted that when we crafted a regimen of combination of carboplatin and a formulation of doxorubicin called liposomal doxorubicin, a dosing strategy that actually is quite tolerated, often doesn't cause hair loss, doesn't cause much nausea or vomiting. It definitely can cause bone marrow suppression, though. We showed there was effect in advanced disease. And then we said, hey, if we treat now early stage triple negative breast cancers with this relatively non-toxic approach, can we see how effective is it? And can we figure out if it's efficacious? Who might benefit from this kind of more gentle approach to the treatment? And so we did a clinical trial in which patients got this kind of combination up front, then went to surgery, and then at the end we of course gave Taxol. So at the end they ended up getting all the agents that are involved in standard therapy of triple negative breast cancer, and then we followed them. And the interesting thing we found was that with this giving half the chemotherapy in a way that was very well tolerated, we had about a third of the patients had a pathologic complete response, the tumor was completely gone. All right. This is still not like with full regimens we can get response rates of over 50%. So this is reasonable. But we said, hey, with this very tolerable regimen, can we identify which patients may do well with this, with this combination? And so right now there's work underway using whole genome sequencing to identify, can we identify which one will respond to this relative this regimen, and can we spare them the more aggressive regimens if they do well long term? All right. And the other thing we're looking at is, okay, what about the tumors that didn't respond? This is work done by Rukia Henry, who's a graduate student in my lab. And we actually made models of these chemo-resistant tumors, what we call patient-derived xenograft models. And we're doing genomics to think, hey, what's causing the growth of these cancers? And can we find ways using a variety of genomic approaches to figure out what's driving the growth of these chemo-resistant triple-negative breast cancers to identify novel ways to treat them? And we hope that this is work that's on progress. We're very hopeful that this may give us new insight into the biology of these chemo-resistant triple-negative breast cancers that are very difficult. And ultimately, we want to sit there and ultimately develop, ways in which can we develop other therapies that target both these primary chemo-resistant and these revertant triple-negative breast cancers, in order to ultimately define combination therapy that may lead to better long-term outcome in women with these repair mutations. And with that, I'll stop. I think we've shown that breast cancer is really made up of multiple distinct diseases. That metastatic disease or micrometastases present even in early stage disease, and that's really the rationale for systemic therapy. That targeted therapy of specific DNA defects, specifically BRCA and BRCA mutations, may improve outcomes both in early stage and advanced breast cancer, and finding rational combinations of therapy based on biology may ultimately, continue to improve outcomes in breast cancer. And we really need to learn more about what drives the growth of individual breast cancers in order to craft treatments that really maximize efficacy and minimize side effects. And with that, I'd like to stop. I'd like to thank all my collaborators. This is work done by a lot of the work that was done here. It was done in collaboration with groups at the NKI, at the Dana-Farber, our collaborators in Europe, as well as investigators at the Cancer Institute of New Jersey and members of my lab highlighted here. And we receive funding from this from a variety of organizations, including the NCI, BCRF, the DoD Breast Cancer Research Program, as well as the LIFE program at the Institute. Thank you so much. We'll be happy to take questions.